Good afternoon to all the EAPCET aspirants. So here we are in our second session of cell cycle and cell division. So we are headed up with, we are done with G1, S and G2 phases of interphase, and we are starting both mitosis and meiosis today, exploring the significance of mitosis and meiosis, and then identifying the differences between mitosis and meiosis. So here if we have to see mitosis or M phase in cell cycle, in complete cell cycle, mitosis or M phase is addressed as most dramatic phase of the cell cycle. Okay. Mitosis or M phase is addressed as most dramatic phase of cell cycle. Fine, right? This is addressed as most dramatic phase of the cell cycle. So here if you see, why is it addressed as most dramatic phase? Because in F phase, as we have discussed, in M phase as we have discussed, we have karyokinesis and cytokinesis. Karyokinesis again has 4 subphases, which are prophase, metaphase, anaphase and telophase. These are 4 subphases under karyokinesis. As this is a progressive process, but there are no clear-cut lines that are drawn in between each substage. Okay, there are no clear-cut lines. Okay, there are no clear-cut lines. No clear-cut lines that are drawn at each substage. Each substage. Clear, right? So here if we see from prophase to telophase, first at prophase what will happen? So once let us recall in our previous session what we have dealt is, in G1 phase maximum cell organelle duplication will happen, cell size will increase and cells will be metabolically active. And in S phase if you see, centriole duplication will take place and chromosomes will get, centriole duplication will take place and DNA replication happens during S phase. In G2 phase if you see, semi-autonomous organelles or self-replicating organelles like mitochondria and chloroplasts will get duplicated, and along with this Golgi bodies will get duplicated, tubulin protein synthesis will happen. These are the activities of what? G2 phase. Now the cell is completely ready with all the 2 sets of organelles, 2 sets of genome material, and it is completely ready with 2 sets of centrioles as well. Now sequential karyokinesis and cytokinesis have to happen. First karyokinesis, that is nuclear separation. Okay. So how does this happen? The first stage is what? Prophase. In prophase, first and foremost thing that happens is the chromatin gets untangled to chromosomes. Okay. Chromatin gets untangled to chromosomes. Fine, right? Chromatin gets untangled to chromosomes and chromosome formation occurs, and the centrioles which are duplicated in the S phase inside the cytoplasm, now these centrioles will move towards the opposite poles. Okay, centrioles move to opposite poles. They will move to opposite poles and they will radiate out the asters. They will radiate asters. Fine, right? Now these asters will be giving out what? Spindle apparatus. The spindle apparatus will be formed, asters will be moved to the opposite poles, and at the end of prophase, that in the late prophase, in late prophase you can see nuclear membrane, nucleolus, endoplasmic reticulum and Golgi bodies will get disintegrated. Clear till here up to prophase? Now you see, now you observe in metaphase what happens is, in metaphase all the chromosomes which are formed, they will get dispersed in the complete cytoplasm and they will get organized into one equatorial plate at the center. Okay, at the center this is addressed as what, ma? Metaphasic plate. metaphasic plate or equatorial plate. Metaphasic or equatorial plate. Fine, right? Now all the spindle fibers which are radiated out of the asters, they will get connected to the kinetocores of this primary constriction. Once you recall the structure of the chromosome, so in chromosome structure, the primary constriction is what, ma? Primary constriction is centromere. To the primary constrictions on both the ends or on the either sides of the primary constriction, you have disc shaped strictures called as kinetocores. To this kinetocores, spindle fibers get attached from the asters, and this will form what? Metaphasic plate or equatorial plate, and this will form two chromosomes, two sets of chromosomes will move towards the opposite poles. Okay, two sets of chromosomes will move towards the opposite poles for two daughter cells. Okay, two daughter nuclei are formed for two daughter cells in the anaphase. So anaphase, in anaphase what is happening? Centromere splitting. In metaphasic plate or equatorial plate, where chromosomes aligned at the center in the metaphase, in anaphase what is happening, ma? Centromere splitting. Okay, centromere splitting. This happens during anaphase. Fine, right? Now telophase, if you observe, telophase, two daughter cells are formed at the end. Okay, two daughter cells are formed, two daughter cells are formed at the end of telophase. Clear, right? So here you see, first is prophase. In prophase what is happening? Chromatin gets untangled to chromosomes, and here centrioles will move towards the opposite poles and they will radiate out the asters. And at the end of prophase, that is in the late prophase, nuclear membrane, nucleolus, endoplasmic reticulum and Golgi bodies will be disintegrated, which means that they are structurally disintegrated. And here if you see the metaphase, in metaphase all the chromosomes are completely spatially arranged in the cytoplasm, and here there is an equatorial plate that is formed at the center where the chromosomes are aligned over the equatorial plate. This is also known as what? Metaphasic plate or equatorial plate. And then in the anaphase what is happening? Centromere splitting happens during anaphase, and telophase where two daughter cells will be formed. Fine, right? Here for this centromere splitting there is a protein that is present called as ACP protein. This will be active during the anaphase. Fine, right? Anaphase complex protein will help in the, or it will mediate the centromere splitting that will be active during the anaphase. This is the complete picture that I can give you for the karyokinesis of metaphase. Now we will move on to the cytokinesis that is happening. Okay, what is cytokinesis? Cytoplasm separation happens in the cytokinesis. Cytokinesis is not the same for plant cell and animal cell. Okay, cytokinesis, which is cytoplasm separation. This is not same for both animal cell or plant cell. In animal cell, if you see, as the limiting boundary is what? Limiting boundary is cell membrane. Cell membrane from the either sides it will constrict forwards. Okay, cell membrane from either sides it will constrict forwards and it will form two daughter cells as such. So the movement here is centripetal movement. Centripetal type of cell division or cytoplasm separation. What is the type, ma? Centripetal, which is from the peripheral ends to center. This is by the formation of cleavage furrow, which is also called as cell furrow. Okay, with the help of cleavage furrow it gets constricted. This is in the from the peripheral ends to the center. So centripetal mode of cytokinesis happens in the animal cell. Okay, centripetal mode of cytokinesis happens in animal cells. If you consider plant cell, you see where at the time of cell division only. At the time of cell division, that is after telophase, what happens in a plant cell is, at the center phragmoplast is formed. This phragmoplast is synthesized from Golgi bodies. Remember, phragmoplast, this is synthesized from Golgi bodies. Later, phragmoplast will get matured to what? Middle lamella. Phragmoplast turns to middle lamella. Fine, right? Phragmoplast turns to middle lamella. Now, this middle lamella will move towards both the ends or both the sides. Okay, middle lamella moves towards the either ends. It will connect at the either ends. Okay, and here middle lamella will be mature to what? Middle lamella will be mature to cell plate.
There is a,
remember, middle lamella is rich in what? Calcium pectate and magnesium pectate. We have learnt in the cell the unit of life at the cell wall segment, wherein middle lamella is composed of what, ma? Calcium pectate and magnesium pectate. Fine, right? Calcium pectate and magnesium pectate. Calcium pectate and magnesium pectate, right? Middle lamella is composed of calcium pectate and magnesium pectate. So, here this gets matured to what? Cell plate. So, it is from center to periphery, right? First at the center phragmoplast is formed, this matures to middle lamella. Middle lamella will touch the either ends forming the cell plate, okay, and as it is from center to periphery, this is addressed as centrifugal mode of cytokinesis, okay. This is centrifugal mode of cytokinesis. Fine, right? So, this is the cytokinesis that happens both on the animal cell and the plant cell. Next, we will move on to meiosis. In meiosis, if you observe in detail, you will have two sets of meiosis, that is meiosis 1 and meiosis 2. Okay. So, here only I will give out the division, meiosis 1 and meiosis 2. Meiosis 1 and meiosis 2, again they will have what? Sequential karyokinesis and cytokinesis. Karyokinesis 1, cytokinesis 1, followed by karyokinesis 2 and cytokinesis 2. So, in between meiosis 1 and meiosis 2, if you see, karyokinesis 1, cytokinesis 1, followed by karyokinesis 2, cytokinesis 2. So, in between meiosis 1 and meiosis 2, there is an interface that is called as interkinesis. Interkinesis. Fine, right? Interkinesis is a stage or intermediate stage that is in between meiosis 1 and meiosis 2. In this interkinesis stage, that intermediate stage, there will not be any DNA replication or duplication. There is no DNA replication or duplication in the interkinesis stage. Whichever proteins and RNA are required for meiosis 2, those are synthesized. Okay, RNA and protein, whichever are required for the synthesis, okay, whichever are required for the activity of meiosis 2, they are synthesized at interkinesis stage, and the centrioles will get duplicated in the interkinesis. Centrioles duplication. Fine, right? So, there is no DNA duplication, RNA protein synthesis will happen, centrioles, if at all they are required, they will be duplicated in the interkinesis region. Okay, so, this is meiosis 1 and meiosis 2. One peculiar stage, that is in the karyokinesis 1, n number of times questions are asked in the EAPCET as well as in the NEET from karyokinesis 1. Okay, wherein karyokinesis 1 will have again 5 substages, wherein the first substage is liptotine, followed by zygotine, followed by pachytine, followed by diplotine, followed by diakinesis. Fine, right? So, you have 5 substages in karyokinesis. 1, that is leptotene, zygotene, pachytene, diplotene and diakinesis. Clear till here? So, this is the framework of what? Meiosis. Let us get started with meiosis 1 first, that is karyokinesis 1. Meiosis always starts from a diploid cell. There is no point of haploid cell starting up the process of meiosis. So, meiosis occurs in what? Diploid cells. The first stage is leptotene. In leptotene, what happens? You see, observe carefully. In leptotene, first thing is, of course, chromatin gets untangled to chromosomes. Chromatin gets untangled to chromosomes. Fine, right? These chromosomes will touch to the walls of the cytoplasm. Okay, chromosomes will touch to the walls of the membrane. So, that is said as spireme stage, or the complete chromosomal thread or chromatid threads will appear as Bocquet stage. Okay, this is also addressed as Bocquet stage. And here centrioles will move towards the opposite poles, radiating out what? Asters. Fine, right? So, what are the events that you have to remember with leptotene? Chromatin material condenses to chromatids. These chromatids will align or touch to the walls of the nuclear membrane or cytoplasmic membrane. So, they are addressed as what? Spireme stage or Bocquet stage. And the centrioles which are present in the cytoplasm, they will move towards the opposite poles, radiating out asters. Till here it is clear, right? So, this is all about leptotene, and one thing you have to remember that the chromatids which are formed in the leptotene are clearly visible under light microscope. They are clearly visible under light microscope. Fine, right? They are clearly visible under light microscope. Next is what? Zygotene. Zygotene is the stage where the chromatids which have same allelic forms arranged at the same loci. Okay. So, if you see, these are the chromatids, chromosomes which have, for example, let us take a character of tallness which is represented by one gene. 2 alleles located at the same loci. So, this you will call it as what, ma? Homologous chromosomes. This you will call it as homologous chromosomes. Fine, right? So, this is known as what? Homologous chromosomes which are formed in the zygotene. So, where does the homologous chromosomes are formed? In the zygotene stage. So, this homologous chromosomes which are formed, they will align to each other, forming up what? Synaptonemal complex. Homologous chromosomes which have same genes aligned at the same loci, they will come together forming up what, ma? Synaptonemal complex. Homologous chromosomes will come towards each other forming synaptonemal complex. So, formation of synaptonemal complex is known as synapsis. Okay. This phenomena is known as what? Synapsis. Fine, right? Formation of synaptonemal complex is known as synapsis. Here homologous chromosomes will be aligned together. Now, here if you see synaptonemal complex formation. So, in a single synaptonemal complex you can see 4 chromatids. Can you see here? 1, 2, 3, 4. 4 chromatids are present in a single synaptonemal complex, and here you can see 2 homologous chromosomes are aligned, right. So, this is known as one synaptonemal complex consists of 1 bivalent and 1 tetrad. 1 bivalent has how many tetrads? 1 tetrad. 1 bivalent which has 1 tetrad. Fine. And here you have to be clear on the note of sister chromatids and non-sister chromatids. Those chromatids which are present on the same homologous chromosomes are known as sister chromatids, and two chromatids of two different chromosomes in the homologous chromosomes, these are addressed as non-sister chromatids. For example, you see here these two chromosomes are addressed as sister chromatids, whereas these two chromosomes from two different chromatids from two different chromosomes known as non-sister chromatids. Chromatids from a same chromosome known as what? Sister chromatids. So, whenever you will get the terms like synaptonemal complex, synapsis, homologous chromosomes formation, or occurrence of a bivalent and tetrad, this and all terms are in relation with zygotine. Clear, right? So, liptotine and zygotine, I hope this is clear to you. Now, we will move on to pachytene. Pachytene, you see here, in pachytene what happens is two non-sister chromatids of a homologous chromosomes, they will undergo recombination. Recombination happens between what, ma? Two non-sister chromatids of homologous chromosomes, they will undergo recombination, and this recombination is also addressed as crossing over. This is not energy mediated process, but this is enzyme mediated process. Okay, pachytene occurs in between what? Two non-sister chromatids of homologous chromosomes. Two non-sister chromatids of homologous chromosomes will get recombined. Okay, this will undergo what? Recombination or crossing over. Recombination or crossing over. Fine, right? So, recombination or crossing over will happen between non-sister chromatids of homologous chromosomes. Clear till here? Fine, right, ma? Non-sister chromatids of homologous chromosomes, they will undergo recombined or recombination. This is an enzyme mediated process. Remember this carefully. This is not ATP mediated process, but this is an enzyme mediated process. And that enzyme is recombinase. First three substages of karyokinesis 1 are clear, no ma? Now we will move on to diplotene. In diplotene what happens? In diplotene stage starts with dissolution of synaptonemal complex. Once after recombination is done, then the synaptonemal complex gets separated from each other. This is known as what? Dissolution of synaptonemal complex is the beginning of the what substage? Diplotene. Okay, this is dissolution of synaptonemal complex. Dissolution of synaptonemal complex is the beginning of the diplotene stage. This dissolution of synaptonemal complex will result in the formation of X-shaped chromosomes. This will result in the formation of X-shaped chromosomes like this. So, the X-shaped chromosomes which are formed, X-shaped structures are addressed as what? Chiasmata. Okay, X-shaped chromosomes or chiasmata. Fine, right? X-shaped chromosomes or chiasmata is formed in diplotene stage. So, here in many cell cycles, like if you consider the mammalian oocytes of vertebrates and invertebrates, those cells are stuck in which phase of cell cycle? They will stop the cell cycle or cell division in the diplotene stage. So, in the mammalian oocytes of vertebrates and oocytes of vertebrates and invertebrates, especially mammalian oocytes, they will stop the cell division at the diplotene stage. So, in those cells specifically mammalian oocytes, this stage is known as dictyotene stage. Okay, in mammalian oocytes, this stage is known as dictyotene stage. Okay, this stage is addressed as what? Dictyotene stage in mammalian oocytes. Till here everything is clear now, pachytene and diplotin. Now, we will move on to diakinesis. So, diakinesis, if you see ma, diakinesis, this stage is the beginning. This stage will begin with disintegration of chiasmata, okay, or dissolution of chiasmata, dissolution or disintegration of chiasmata. Fine, right? And this is followed by what? This is followed by nuclear membrane, nucleolus, endoplasmic reticulum and Golgi bodies disintegration. This is diakinesis. Clear, right? Which is the last step of karyokinesis 1. Now, at the end of karyokinesis 1, you will have two sets of chromosomes which are recombined. Okay, two sets of chromosomes which are recombined like this. These two sets of chromosomes are aligned in bimetaphasic plate. Okay, they are aligned in bimetaphasic plate. Means you see two sets of chromosomes are aligned in two equatorial plates. Okay, so the sister chromatids of each chromosome are present in one metaphasic plate. And how they are connected to spindle fibers ma? With the help, spindle fibers are connected to kinetocores of the chromosomes. Okay, on the either sides of two sets of chromosomes, the kinetocores are connected to spindle fibers. As such, the bimetaphasic plate is observed. Okay, bimetaphasic plate is clear, right? This is in metaphase 1. Now, in anaphase 1, if you see anaphase 1, what happens is two chromosomes are moving towards the opposite poles, right? Two chromosomes moving towards the opposite poles, and here in anaphase 1, ACP protein, which is ACP1 protein, which is responsible for centromere splitting, is inactive. Okay, and there is no centromere splitting here. No centromere splitting. Okay, here there is no centromere splitting and ACP protein is inactive. As such, the sister chromatids of each chromosome move towards the opposite poles. Remember, what is moving towards the opposite poles ma? Sister chromatids of each chromosome moves to opposite poles. Fine, right? Next, last is what? Telophase 1. Okay, in telophase 1, two daughter nuclei are formed in the anaphase 1 itself. So, in anaphase 1 only the ploidy gets into half. The ploidy gets half in the anaphase because two sister chromosomes are moving towards the opposite poles, and here chromosome number will get reduced to half. Ploidy reduces to half at the end of anaphase 1. And in telophase 1, if you see, the chromosomes will get tangled to chromatin again, and here you can see the formation of nuclear membrane, nucleolus, endoplasmic reticulum and Golgi. They will be reformed again at the end of telophase. Till here everything is clear, right ma? Anaphase and telophase 1. Now we will move on to meiosis 2. Meiosis 2 is same as that of mitosis. Mitosis, as there is no ploidy reduction, as there in no point you can see chromosomes are reduced to half. The chromosome number of a parent cell is same as that of daughter cell, right? And mitosis occurs in somatic cells, as there is no chromosomal reduction, mitosis is also addressed as what? Equational division. Whereas in meiosis you can see here in anaphase 1, just now we have seen, wherein ploidy gets reduced and chromosome number reduces to half. Here in meiosis 2, okay, in meiosis type of division is addressed as reductional division, okay, meiotic division is what ma? Reductional division. And here you see meiosis 2, prophase 2, metaphase 2, anaphase 2 and telophase 2. This is same as that of mitosis, same as that of mitosis. So, in prophase 2 what happens? Chromosomes are formed, chromatin again gets condensed to what? Chromosomes, and here centrioles will move towards the opposite poles. That and all our events are happening in prophase 2. And in metaphase 2 if you see, there is single metaphasic plate that is formed, same as that you see in the mitosis, okay, single metaphasic plate, okay, single metaphasic plate is formed at the end of metaphase 2. And in anaphase 2 if you see, ACP protein is active in anaphase 2, okay, ACP is active in anaphase 2 and centromere splitting happens in the anaphase 2, centromere splitting happens in anaphase 2. Next in telophase 2, 4 daughter cells are formed, right. So, two daughter cells which are formed at the end of meiosis 1, they will again get duplicated in meiosis 2. As such, 4 daughter cells are formed, okay, 4 daughter cells are formed at the end of telophase, 4 daughter cells are formed, clear, right, clear till here. Now, we will move on to the clear differences between mitosis and meiosis. What are the differences between mitosis and meiosis? Here we go. First, mitosis, as we have discussed, these occur in somatic cells, whereas meiosis occurs in germ cells. Fine, right? Here nucleus divides only once in mitosis, nucleus divides twice in meiosis. 2 daughter cells are formed in mitosis, 4 daughter cells will be formed in meiosis. Daughter cells ploidy is diploid in mitosis, daughter cells ploidy is haploid in meiosis, because here chromosomal reduction is taking place. It occurs more frequently, mitosis occurs more frequently, whereas meiosis occurs less frequently. And here daughter cells will form somatic organs, here daughter cells form gametes, daughter cells form what? Gametes. And here there is only one prophase, one metaphase, one anaphase and one telophase. Here you can see there are 2 of each phases. You can see there are 5 subphases in prophase 1. We have discussed what are they ma? Leptotene, zygotene, pachytene, diplotene and diakinesis. These are the 5 substages in prophase 1, and there is no crossing over here in the chromosomes because there is no pachytene in mitosis. Crossing over or recombination happens in meiosis. This is also addressed as reductional division and this will be equational division. This is complete. In brief, I can tell you about mitosis and meiosis. Here what all are important is the substages specially of meiosis 1 and the ploidy. Most importantly, what are the events of each phase of the M phase, that is important. So, in our next class, we will deal with the significance of mitosis and meiosis and we will exercise the previous year EAPCET questions from mitosis and meiosis, and then we will start the chapter biomolecules. So, come prepared with the chapter so that we can exercise the questions more brightly and more confidently. That is all for today's session ma. Thank you.